Hi, Ms. Patterson here. Today we're going to go over organelles for AP Biology Unit 1. This is going to be a longer video because I have to go through so many organelles. So up here I'm going to do timestamps of each organelle. So if you want to stop, look for a certain one, jump to that time. If you want to come back in another day to look at some other ones, you're more than welcome to. So let's first talk about two types of cells. I have prokaryotes and I have eukaryotes. When you see prokaryotes, you can see they have a nucleoid. A nucleoid is a circular DNA strand inside. I have ribosomes, which are these small little dots right here. These are going to make proteins. I have a cell wall. I have a capsule. I have a flagellum. And I'm going to have a cell membrane that's all going to be part of this prokaryotic cell. Eukaryotics, just looking at it, you can tell that it's going to be a lot more complex. I have more organelles. I still have a cell membrane. I still have ribosome, but I have mitochondria. I have an enclosed nucleus. I have a cytoplasmic membrane. I have cytoplasm. I've got all of these more complex organelles. There are characteristics that are specific for prokaryotic and eukaryotic cells. We have a plasma membrane. Those are going to be in both. All cells are going to have a plasma membrane. Ribosomes are going to be in both. Ribosomes are a very primitive form of an organic molecule, and this is going to not have a membrane bound outside. This is going to be kind of one of your more basic organelles. You have membrane-bound organelles. Those are only going to be found in eukaryotic cells. A nucleus. Prokaryotic cells, they have a DNA strand. It's circular, but it's free-floating in the cell. Whereas a eukaryotic cell has a true nucleus. It's going to be enclosed. It protects the DNA. The size for a prokaryotic, it's small. It's about 1 to 5 micrometers. Where eukaryotic cells are a little bigger, they're between 10 and 100 micrometers. Organelles, there's 3 types we need to look at: non-membrane bound, the endomembrane, and the energy organelles. So here you can see this is a eukaryotic cell. I have all of these organelles that are here. Non-membrane bound organelles, do you remember which ones those are? Those are my ribosomes. So ribosomes are these tiny little dots you see here inside the cell. These are going to be where protein synthesis happens. Whenever I have mRNA going to go in there and going to make ribosomes or make proteins from ribosomes. So the ribosomes are going to be non-membrane bound organelles. Your endomembrane, endo means inside. So your endomembrane organelles are going to be the ones that are going to have some type of plasma membrane. So your nuclear envelope, which goes around the nucleus. Endoplasmic reticulum, which is kind of like this maze-looking piece. You have the Golgi complex. Sometimes they'll call it the Golgi Apparatus. This is part right here. You have lysosomes. Those are going to be this part right here. And then you have vesicles, which are going to be some types of like little tubes, little bubbles that are kind of like inside the cell. Plasma membrane is this outside piece right there. Energy organelles, you got 2. You got mitochondria and chloroplasts. They're going to have a membrane on the outside, but it's a double membrane. And we'll talk more about those when we get to the energy organelles. So compartmentalization, this is pretty big. This is going to make sure that the organelles inside the cell aren't interfering with reactions that are happening. So I have lysosomes. These are going to not interact with mitochondria. Mitochondria won't interact with anything else going on. These are going to be very specific and very specialized. Think of this kind of like a fishing tackle box. If I have a tackle box, I'm going to have a spot that's just for hooks. I'm going to have a spot just for weights, a spot just for bait. And they're not all going to mix together. I don't want them to mix together. Same thing you could think if you have a tray of paint. And my paint's going to be in different compartments. And I don't want to mix those paints. It's not going to be a good thing. So same thing with organelles. They have compartments that are going to be very specialized for what they need to do. I can increase surface area for these reactions. So here you see these mitochondria, how I have these little pieces that kind of come in and come out. This has increased the surface area for mitochondria for me to be able to make more ATP. Here you see the rough ER. All these little dots are little ribosomes. And so there's going to be places where proteins can be made from those ribosomes. And the more surface area I have on this ER, the more proteins I'm going to be able to make because I'm going to be able to hold more ribosomes. Here are two types of cells. I have plant cells. I can tell because it's a brick block shape. Animal cells are going to be some type of a regular or round shape. And there's characteristics for those. So plant cells, animal cells, with lysosomes, I'm not going to have any in plants, but I'll have them in animal cells. Now, plants can have a type of lysosome. Sometimes vacuoles can be a form of lysosomes. So when we talk about lysosomes, I'll try to remember to bring that back up. But plants don't have true lysosomes, but some vacuoles can act like lysosomes. Chloroplasts are going to be found only in plant cells. Centrosomes are only going to be found in animal cells. Flagella is going to be found sometimes in animal cells. Central vacuole will be found in plant cells. Animal cells will have a small vacuole, but they're going to be small, kind of like these little white dots right there. They're going to be kind of like small little spots. They're not going to be as large and take up as much volume as a plant vacuole. A cell wall is going to be in a plant. This is going to be this part right here. And the plasmodesmata will also be in a plant cell. These are going to be openings in this wall that are going to allow material to pass from one side to the other. I'm not going to see those in animal cells. Okay, the plasma membrane. This is a super important cell organelle. This is going to be found in both plant and animal cells. It's going to be made of phospholipids. So if you remember what kind of macromolecule would be a phospholipid, right there is your clue. It's going to be a lipid. So in your phospholipids, you have a hydrophilic head. This is water-loving. A hydrophobic tail, which is water-fearing. And I have a phospholipid bilayer. So bi means two. So I have two layers of these phospholipids. And these non-polar tails are attracted to each other. Because remember, all this outside is water. And these don't like it, so they're going to fold in towards each other. These hydrophilic heads are going to go out into the aqueous solution. Outside the cell, inside the cells, kind of a water solution. And this is going to be very selective. This is going to allow material in and out and be very selective on what it allows to move through, what it makes leave. It's going to be very selective with that because of this membrane. This membrane will be on the outside of the cell. This is also what kind of surrounds other organelles within the cell. So here I have a cell wall. The cell wall is part of a plant cell. I can see all of these little blocks. All of these blocks represent those plant cells. They're going to be protecting the inside of this nucleus, the inside of this chloroplast. It's going to have a thick wall that's going to prevent dehydration from the water. It's going to help shape it. So these blocks like to lay on top of each other and they're going to get real tight, kind of like Legos. This is going to allow support for this plant to be able to stand up and do things that it needs to do. It's going to be made of cellulose. So if you remember which macromolecule is a part of cellulose, it's going to be a carbohydrate, and it's one of the ones that are insoluble in water, which means I cannot dissolve in water. And if you remember, humans can't eat cellulose. You have the alpha glucose and the beta glucose, and they twist. So your bond's up and then your bond's down, and we don't have the enzymes to break down those specific bonds. So we could eat grass all day long and we would starve to death. But plants are able to use this to an advantage and use it as part of a support system for their cells. We have plasmodesmata. These are little holes. You can think of them kind of like doors that are here inside the cell. If I need to get material from this cell to this cell, I can't just not move stuff through if these were solid walls. So they have to have openings. So they're going to pass through, pass through, pass through, pass through to get to wherever I need to go. So here's an example. These lines here, these are a cell wall. These little openings are the little doors. These are your plasmodesmata. When you see this curved part right here, this is the plasma membrane. So remember, all cells have a plasma membrane. A plant cell's plasma membrane is inside that cell wall. And so it's selective. It's going to allow material back and forth through this plasmodesmata. But if I need to get material over here, I can pass it through, pass it through, pass it through to get wherever it needs to go. The nucleus, this is going to be this whole part right here. They're both in plant and animal cells. I have DNA inside. And DNA, remember, it codes for proteins. And RNA is going to help with protein synthesis. It's going to have a nuclear envelope. So you see these double lines right here. This is a plasma membrane, but you notice I have two. So this is a double membrane. So I have one on the outside and one on the inside. So that phospholipid bilayer that we looked at, I have two. I have one here and one here. So this nuclear envelope is going to house all of this. It's also continuous with this rough ER. So it's going to be attached to this. If I'm making RNA and messenger RNA needs to go to ribosomes to make proteins, I'm going to try to get it as quick as possible. So you see all these little dots? All these little dots are ribosomes. And these are going to make proteins from the mRNA that's coming out. So the RNA comes out, it's going to hit this protein, or this ribosome, make some proteins, and it's going to send it out. So this is a very fast way to do it. It's very convenient to have all of this right here. You have nuclear pores. So see all these openings? These are going to be very selective, what it allows in and out. It can let mRNA out. It can let material in. These little flower petals here are pores too, and it's all surrounding this nucleus. Chromatin are these little hair-like string structures. These are kind of condensed forms of DNA. So let's talk about that. I have a strand of DNA. I have a complementary strand here, and here's my DNA. It's a helix, which is like a curved piece of material. And you see these spheres right here? These are protein spheres. And DNA just doesn't wrap around itself. It's going to wrap around these histones, these little protein spheres. And it's going to curl together, and it's going to create what's called a nucleosome. And it's going to curl and curl and curl. And it's going to resemble something kind of like the old telephones that have the cords that are like curlicues. And it keeps the cord from getting tangled. And that's what these histones do. They kind of prevent this DNA from getting entangled amongst itself. These nucleosomes are going to curl up tight, and these are going to make your chromatin. So those little string things that we saw inside the nucleus, this is the chromatin. And it's going to be relaxed. And then once it gets ready to start to divide, it's going to start to tighten up and turn into these chromosomes for cell division. So here I have this chromatin. This is those condensed little strands right there. The nucleolus is this part right here. It's the dark part in the center. All of this here is the nucleus. The nucleolus is right here. This is the part of the nucleus where our RNA gets synthesized, where your ribosomes kind of get made, is right in here. So your rRNA are your two subunits of what make a ribosome. Ribosomes, this is what that is made into. It's two parts. I have a large subunit and a small subunit. They look kind of like little hamburger buns. And these are your protein factories. These are going to bring in mRNA. And that mRNA is coding for a protein. tRNA is going to come in, pull an amino acid, and I'm going to make it an amino acid sequence. And this is going to turn into a protein. It's going to be composed of RNA and proteins. So I have RNA and I have proteins. Okay, all of this is going to be your ribosome. It's going to be protein synthesis, large and small subunit. And when the protein goes away and the mRNA leaves, it kind of detaches. It's going to fall apart, and then it's going to come back together when another mRNA strand comes in. You have two types. You have free ribosomes. These are ones that are just going to be floating in. So these little dots right here, these are my free ribosomes. They're going to be producing proteins. They're going to be producing material for the cell to be able to use. You have bound proteins, so these ones here on the ER, you see how all of those ribosomes are attached to the rough ER. These are going to make proteins that are destined to leave the cell. So they're going to get packaged, they're going to get modified, and then they're going to leave the cell to go someplace else. So here we have endoplasmic reticulum. We call it the ER. This is going to be in both plant and animal cells. So here's the nucleus. Here's my rough ER. This is where all my little ribosomes are. And then here I have what's called a smooth ER. So it makes up about half the total membrane structure of the cell. This is pretty big. This takes up a big chunk of the cell. So this is going to make membranes. You're going to have membranes also on the outside. Remember, all your organelles have a membrane on the outside. So your two types, you have your smooth ER, which is this one right here. I do not have ribosomes on it. That's why it's considered to be smooth. And this is going to be the synthesis of lipids. I can make lipids with this smooth ER. It also helps to detoxify. So if you have drugs, certain types of poisons, it's going to go in there. It's going to help detoxify, break it down. The other one is your rough ER. This is bound ribosomes, which are these ones right here, these little dots. They're going to make proteins that can be excreted from the cell. So you see here, this little spot right here says transport vesicle. This is part of this rough ER. And there's something on here that needs to be shipped out. So this is kind of like, you think of this membrane like a bubble. If I took a bubble and I had a big bubble, I could break it and I could kind of like mess with it and I could pull a small bubble off. And that's kind of what's happening with the rough ER. There's a small bubble that gets pinched off, and it's going to have the material that was made, the protein that was made by these ribosomes is going to go in here. When it goes into this vesicle, it's going to go to what's called a Golgi body, and we're going to talk more about that here in a second. But this is how it travels through the cell, is through these little bubbles, these little vesicles that are membranes. I have a phospholipid layer here. This membrane is going to allow it to go through the cell without being destroyed or broken down. So it's going to have to go to the Golgi body. So once it leaves the rough ER, it's going to go to what's called the Golgi body or Golgi apparatus. This is also the membrane factory for the cells. So all this membranes for a lot of the organelles in here for the outside of the cell is going to make it through the rough ER. All right, the Golgi apparatus. So I'm getting the vesicle from the ER, and I have a directional way to move. So these proteins are going to come, and I'm going to manufacture certain macromolecules from this ER. I'm going to modify, I'm going to ship it, and it has these flat little sacs, and they don't connect. They're like little pancake sacs. So the vesicle goes from the ER, and it comes in through what's called a cis phase. It's directional. I have to go in through this cis phase. This vesicle outline, this membrane, is going to fuse with the Golgi membrane, and it's going to become like one bubble. So if I have one bubble and one bubble and I put them together, I can sometimes make one big bubble. And that's kind of what it does with this membrane. The membrane is going to fuse with this. When it does, it's going to deposit whatever protein was made in here. There are enzymes, there are other proteins in here that can get added onto this protein. Stuff can get taken off. I can add tags to this. I can change it up in a way that's going to make it more functional. So I'm going to go inside that cis phase. It's going to be the receiving end. It's going to come out what's called the trans phase. This is the part that comes out. When it comes back out, you see that I have a new membrane. So now I've turned into another transport vesicle that is made from this Golgi membrane. So this piece has now been modified. It's been changed. I've added or taken stuff off. And now I'm getting ready to go outside the cell. So this is going to go. It's going to fuse with this plasma membrane. And you can see how it's all one piece now. This is how some membranes can grow, get smaller. This will attach, and I can enlarge my membrane from these vesicles. It's specialized for cells to be secreted, so they're going to be made to leave the cell. Lysosomes are going to be found in animal cells. I'm not going to find them in plant cells. Now, some vacuoles can act like a lysosome, but plant cells do not have a true lysosome. Animal cells do. they're going to hydrolyze. So what does hydrolyze mean? Remember, hydro means water, lyse means to cut, and when we talk about our macromolecules, we talk about hydrolyzing those bonds, and that means to cut them. So hydrolyzing enzymes, these are enzymes that are going to start cutting material apart. You have digestive molecules, and it's called phagocytosis. I can bring in food particles, and they're going to be in those vesicles. Remember, they're like those little membrane-bound bubbles. And these bubbles are going to come in, and they're going to attach to a lysosome, which is kind of like a bubble that has very specific hydrolyzing enzymes inside. They're going to start breaking stuff down. So you can see here the lysosome is attached to this vesicle. It's going to excrete this enzyme into it. It's going to become one big bubble, and you can see how it starts to break it down. Then the lysosome is going to take it wherever it needs to go. If it needs to be reused, it can recycle it. If it needs to be pushed out of the cell, it can go do that too. You have autophagy, and this is when it's going to recycle what it needs. And sometimes it's mostly organic molecules. If it needs to use material to make more carbs, if it needs to make more lipids, it can break down those pieces, save what it needs, get rid of what it doesn't, and recycle it for the cell to be able to use later. If some cells start to get wore out, like if a mitochondria starts to not function correctly, it can go in and it can start to break down that mitochondria, recycle pieces that it needs, and get rid of the pieces that are not useful. Hydrolyzing the enzyme works best in an acidic environment. So if these lysosomes burst inside the cell, that doesn't mean that it's going to start tearing everything apart. This doesn't work good in a neutral pH, which is what a cell is. So it kind of deactivates this lysosome from breaking things down. So a lysosome enzyme inside is a pretty acidic pH. This is also a really good example of compartmentalization. This is going to be very specified, very specific to one type of function, and it does it very well. Okay, the next one's a vacuole. So I can find that in a plant or animal cell, but they're going to look different in both. So you have food vacuoles. Those are going to be ones I'm going to find more in animal cells. They're small, they're able to move, I'm able to store food, use them for later. So phagocytosis would be an example of that. So phago means to eat, cytosis means like of the cell. You have contractile vacuoles. These are ones that are going to push to regulate water. So you can see this little hole right here. This is a paramecium, and water will come in and it will have to expel it, so it will squeeze out water in order to make sure it stays balanced. You have a central vacuole. You'll find those in plants, and you can see how large this vacuole is compared to the one that we have in animal cells. This is going to allow what's called turgid pressure, and this is going to allow the plant to stay upright. So when a plant starts to wilt, this vacuole has lost water, and then when you water it, it fills back up and the plant will perk back up. And so this is kind of allows the plant to have a little bit of pressure inside for structure. This is only in plants. This is going to take up quite a bit of space in the plant, around 80% of the volume. All right, the next one is peroxisomes. These are specialized enzymes that can remove hydrogen atoms. These are going to be from various substances, and they're going to transfer them to oxygen. So here I have a peroxisome right here. You can see they're kind of small. They're about the size of lysosomes. And they're going to break down material, and it's going to form hydrogen peroxide. Now, hydrogen peroxide is pretty toxic to the cell, and so it's able to go in and use that. So it uses oxygen to break these fatty acid tails. They're going to send it to the mitochondria to help make ATP. And ATP is the energy currency of the cell. In the liver, it's going to detoxify alcohol and other compounds that could be harmful to people. So it's a different type of organelle that is very specialized, more specifically, but it can be extremely toxic if it's not done correctly. So here I have mitochondria, and this is with mitochondria. This is if I have an electron microscope. This is what it looks like underneath that. I can see these little curves, that I have inside of this mitochondria drawing here. This is going to be the site of cellular respiration. These little grooves are going to increase my surface area, so I have more areas to make ATP and for cellular respiration to happen. I have a double membrane, so on the outside here you see I have a membrane. This little orange part here on the inside is my inner membrane, and this is going to curve. This whole little jelly bean thing in the middle is what's called a cristae. You have matrix, this is the gel-like substance inside. And then you have ribosomes. So see these little dots? These little dots are your ribosomes, and these are like bacterial ribosomes. These are more primitive than the ribosomes you see in a eukaryotic cell. Your mitochondrial DNA is also different. Mitochondria has its own DNA, and it's circular, so it's similar to like a prokaryotic DNA. It's like a nucleoid. It has a circular shape to it. And what's interesting is this DNA is maternal. So this DNA is passed down through the women in your genetic heritage. So your mom's DNA is in these mitochondrial DNA. So it's very interesting that it has its own DNA and it's passed down through the maternal side of your family. Here we have chloroplasts. These are going to be another energy-making cell. These are only found in plants. This is going to be the site of photosynthesis. And this is going to be the process that's going to bring in sunlight, and it turns it into chemical energy form of glucose. It has a double membrane. So here on the outside, I have this double membrane. I have an inner membrane here on this side. And I have membrane-bound components. I have these little, I like to call them like little Oreo cookies. They look like little Oreo cookies. These little Oreo cookies, these are little thylakoids. And then a stack of these is what's called a granum. And then all around this, there's a fluid, and it's called the stroma. And so the stroma kind of covers all of these thylakoids. Here you have what's called a thylakoid membrane. So this is that thylakoid. If I cut it open, it's got an open spot inside, and it's the thylakoid membrane. This is kind of where photosynthesis happens. I have lumen inside, which is like a gel-like substance that's inside of these thylakoids. So the thylakoids are super important for photosynthesis. My stroma, this is going to contain chloroplast, the DNA. Chloroplasts have their own DNA too. They're circular, and they have their own ribosomes, and they're the type of bacterial ribosomes similar in mitochondria. So there's a theory, Endosymbiotic Theory, and this is that all of mitochondria and chloroplasts have descended from some type of bacterial cell, some prokaryotic cell from long ago. So this cell has a circular DNA, and then there was once a cell that was very similar to mitochondria. It also had a circular DNA. The evidence that we say, hey, this is true, like we believe that this was once a bacteria, is that it has a double membrane. When you look at prokaryotics, they have a double membrane. They have a capsule and a membrane. It contains free ribosomes. All your ribosomes are floating free inside of this mitochondria or chloroplast. I have circular DNA, which they both have. And they grow and they reproduce independently. I don't have to have the cell tell the DNA to replicate in a mitochondria or in a chloroplast. It does it on its own. So mitochondria is non-photosynthetic. So it's believed that this mitochondria got absorbed into another cell. And instead of the cell breaking it down, the cell recognized that, hey, it gives me energy. I'm able to use this, and in turn, I can give this mitochondrial bacteria something in return. So they have a symbiotic relationship. They're able to help each other out. So the cell absorbed the mitochondrial bacteria, and it ended up incorporating it into all of its cell functions. Later on, we have the chloroplast come along. This is photosynthetic. So sunlight is going to trigger it to make sugars. The cell is going to take it in, and instead of destroying it, it realizes that they can work together. They have a symbiotic relationship, And that's kind of the idea behind why we have two types of cells, organelles, inside these eukaryotic cells that are independent and they don't act like all the other organelles. So now I have structural networks. There's three types. So these are our cytoskeleton networks. You have them for support, you use them for motility, and you use them as anchors. So I have different functions for all of these cytoskeletons. So you think cyto means cell, skeleton means support or structure. So this is going to give you all of those inside this cell. So there's three main types. So let's talk about the first one. The first ones are called microtubules. These are going to be the largest cytoskeleton fiber. They're going to shape and support, and they're going to provide these tracks for what are called motor molecules. And these are kind of neat little things. They're kind of like little robots that walk on these tubes. So you'll have a long tube, and then these little motor molecule, this protein, is going to kind of like walk along this little tube. So let's say I had to get something from the rough ER to the Golgi. There's going to be a microtubule that is going to kind of connect. It's going to make it like a little track that's going to allow the material, my cargo that I need to have over here, to move to over here so I can go in the cis phase and get modified. So this protein is going to come along and it's going to carry the cargo, and it's just going to walk it down that tube and take it where it needs to go. Then it will drop off the cargo, and then it can walk back and get some more, and it can just keep using this little path to move back and forth. It can separate chromosomes during mitosis and meiosis. So you'll see these when we do diagrams. You have these centrioles, and these centrioles are microtubules, and they can produce these tubes that can come off, and they'll hook and they'll pull chromosomes back, or they can move them around and adjust them inside the cell. Here I have a centriole, and you can see I have these microtubule triplets, 1, 2, 3. These tubes are going to make a circle, and these are going to provide some support for these centrioles, these organelles. I can have structural components of cilia and flagella. So flagella are kind of like these little tails. They're like tubes that are kind of like little whips that can move organisms through water. You have cilia that are small little like hairs that will kind of help to sweep, and they call them, they beat. And so they beat and they move and they sway, and they'll move in certain ways. Now what's interesting about flagella is they're made of these microtubules, but they're covered in a plasma membrane. And so you see how these tubules are in twos. These are called a doublet, and these will kind of go together and they'll make a circle. I'll have a circle of them, and then I'll have two of these microtubules here in the center. You see these little yellow spots right there? Those are some of those motor protons or motor proteins again, and they're going to start walking. And when they walk, that's what kind of causes the flagella to flip and to move and to undulate inside that fluid. So here's a cross-section of an electron microscope of a flagella, so you can see these tubules right there in the center. Another one is microfilaments. These are going to be made for the protein actin. These are small little pieces of protein that are going to allow things to be pulled together. And these are used when we do muscle contractions. So here I have this little bar and this little bar, so this is like a cell. And then I have actin, which is my microfilament. And these are going to have like little holes. They're kind of like little twists. And you see how I got these little holes inside of here? I have myosin, which is another type of filament, and these are kind of going to be like little hooks. And they're going to hook inside this actin. And as it hooks, it's going to keep hooking back, and it's going to pull back this way. And this one's going to hook, and it's going to pull back this way. And this is going to cause this muscle to contract. So you can see how I was here, and then I was able to pull this actin here, pull this actin here. And when I want to release it, then they'll unhook and they'll kind of push back, and then I'll go back out. So this actin will pull on those myosin filaments to pull it in, and that causes a contraction. Intermediate filaments, they're not as exciting as the other two, but they're needed. These are going to help maintain shape, and it's going to fix position of certain organelles. So you need to think of it like a tent. If you go camping and I put a tent out on the ground, it has no shape. So what gives a tent its shape? It's going to be these poles and these cables that are going to pull on it to give it a very specific shape. So these intermediate filaments are a great example of that. A good example is a nuclear lamina. This is going to be the outside of the nuclear membrane, and that's going to give it the shape of the nucleus. So here I have the lamina, and here is going to be my intermediate filaments. This is going to give that shape, that structure for that nuclear lamina. So here I have intermediate filaments. These are going to help anchor. These are going to help support. I have microfilaments. I have microtubules. And so these are all working together on some parts of the cell, and some will work independently. Here I have some more flagella. So these are long tails. Sometimes they can have a lot. They can have a couple. They're going to help propel. These are going to be for movement. So I can have ones with one tail. I can have some on both sides, one on both sides. I can have multiple tails. So you can have flagella that are very, very different. Sometimes they call them flagella. You'll hear flagella. It's the same thing. Cilia, these are the little hairs that are on the side. These will help with movement. They can help sweep to push food inside mouthpieces. They're really good at doing that. You also notice them inside your trachea. So you have these cilia. So whenever you get sick and you start coughing, your cilia inside your trachea is constantly pushing stuff out. And so when you have a buildup of mucus, you have a buildup of some type of material, you cough, and you're coughing to try to clear that. And so it's the cilia constantly pushing and sweeping the stuff out of your trachea. All right, that is it for right now. So that was organelles. That was a pretty long one. If you guys have any questions, let me know, and we'll talk to you later. Bye.